Well, thank you very much, Joanna and Angela, for having me today. And hi, everyone. So I am a scientific engagement manager for protocols.io. And so it sounds like quite a few people on the call are maybe not familiar and haven't used protocols.io. And what it is a workflow tool specific to research methods and protocols. So for my presentation today, I'll start talking a little bit about kind of the history of methods and maybe frustrations that researchers have encountered when trying to find methods. And then I'll talk a little bit about kind of this shift that's happening towards new ways of working and sharing methods openly. So let's get started. So if we go back a little bit in history, but not very far, only back to 2004, that is when the journal Nature Methods started. So quite recently, actually. And at the time, there was a lot of skepticism, even in the Nature publishing group, around launching this journal, because the idea was that they publish research, no one's going to read only methods. It's, impossible to fill a journal's pages with only methods, and so on. But I think, um, if we fast forward to now, something like Nature Methods was a huge success. It continues to be a huge success. And I think it's quite interesting to think about the skepticism that was happening at the time around giving methods independent credit. And so now this skepticism seems a bit surprising because publishing methods can be very impactful. So back in 2014, and then again just last year, the Nature Journal compiled a list of the most highly cited papers. And if we look at the list there, um, the most cited papers of all times are dominated by biological laboratory techniques. So things like techniques and methods. And each of these has over 200,000 citations. So what this is showing us is that methods are very important and they do have a big impact. If we think about what's kind of happening in research generally, is that one of the things we still see is that outside of, publishing a, you know, finalized paper in Nature Methods, what about all of the methods that go into research papers? And there's a spectrum, but we do see things like this shown on this slide. So these are some examples from researchers complaining on Twitter about their frustration over the lack of detail in methods published within research papers. So my personal favorite is the one in the middle, where we have a citation chain where it says devices were fabricated as previously described, and then finally, when you get back to the original publication, it says devices were fabricated with conventional methods. So that information is not very helpful, and it's not going to be useful for someone if they want to try and build upon that method and advance science. And so what this causes when we have a lack of information is overall um, a lack of reproducibility. So this is an example from quite a few years now. Back in 2017, we can see this here on the left was a write-up that was done about this cancer reproducibility project in the Atlantic. And I think the key thing here is that, they tried to reproduce these very 50 high impact papers in cancer research. They were unable to do that. The project was stopped because they could not reproduce these papers. And one of the key reasons for that was that the recipes or, methods and protocols from these papers were incomplete, missing important steps and details, or weren't described at all. And then on the right here, we can see some more statistics about this project. And so you can see, again, there 0% of the protocols were completely described. This is another more recent write-up that was done on the topic of reproducibility from last year. And I think, with reproducibility, one of the things is that scientists care about reproducibility, but there are, changes in behavior that need to happen. And it's not because people don't care, it's because everyone's busy and other things become a higher priority. And so I think this is a really nice quote from this article that was published. And so if we start at the bottom, actually, we can see that, these problems demand action, such as open sharing of data and protocols, which I think, can absolutely help improve reproducibility. But what I do want to highlight here is what we can see above it, which is this researcher saying that low reproducibility highlights structural weaknesses in the way we produce, document, and communicate science. So reproducibility doesn't start at publication when you're trying to communicate your science out. It starts in the lab while you're doing the experiment. So in that produce and document stage. One other thing, excuse me, that we can look at is how people are sharing their methods currently. And so, lots of people are doing a really great job of that. And so these are some of the things that people are doing. So one of them is, putting method details into the supporting information of journal articles. This is a great first start, but there are a few drawbacks of that approach. One of them is that supplementary information is putting that information in a static PDF document that is not, built for a method or a protocol. People can put that information in a variety of ways, so there's no standard format. And then another important thing is that it's a lot less discoverable when it's in the supplementary information because, you know, search engines don't kind of pick up that text. It's not text mined, so you can't search for it independently of the research paper. And that means if you have a method that might be applicable across a range of disciplines, it might not be found from people in other disciplines because it's only attached to a specific paper from a specific discipline. And we'll see an example of that a little bit later on. What people also do, and is again a really great start, is sharing lists of the protocols that they use in their lab on kind of individual lab websites. So there is time and effort that's needed to kind of get these websites set up and running. But what we see happen here is that there is a problem with persistent links. So these links to these websites, they break over time. So people might move from one university to another, or the lab head changes, they stop being maintained over time. And so if someone is using these protocols, is citing these protocols within a research paper, if you follow that link 10 years down the line, it might not give you the paper anymore. So we need more kind of persistent links that are maintained over time. So something like a DOI. So I want to highlight as well, we've talked a lot about reproducibility, but reproducibility isn't only about other people trying to reproduce or build on your work. Most of the time, the person who will be reusing and building upon your own work is you. So again, everyone has different kind of styles and methods of documentation, but when they're busy, sometimes they can slip. And 6 months down the line, when you're wanting to share your methods later, you might not always remember what parameter changed, what was the best version of a protocol, things like that. And so when it comes to sharing methods openly, it can be a lot of work to get all of that information together to share them out because you're trying to reconstruct kind of decisions that were made a long time ago, and that information might be scattered across multiple pages, a paper notebook, online files, emails. And so that's where that time and effort gets deprioritized over other things, that might be more important, getting that next grant, things like that. But really, and what these kind of screenshots show on the slide here is that if you do put a little bit of extra time in while you're doing the work, it can save you a lot of time in the long run. And so I think if you think about documenting your methods clearly while you're doing them, you're going to save yourself time when you build upon your work. And then you're also going to save yourself time when you come to share those methods openly because that documentation work has already been done. And there are tools that can help do this. So protocols.io is one of them. And so what I'll show shortly is that there are two sides of the platform that can enable that. So one of them is a way for you to help you document your methods as you're working. And then the downstream effect of that is that you can push them public and make it easier for other people to build upon your science. So these are the two sides of protocols.io. And so we'll start on the left on the private side of the platform. And so that's where we're an experimental workflow tool where you can document and track your own method kind of tweaks and evolutions over time. So you can do that on your own. You can also do that in a collaborative space with your lab, or if you do collaborate with people in other places as well. We also have, various tools to help you do your work, to make it easier to draft your protocols, troubleshoot them when they're not working as well. So the idea with protocols.io is that you set up your system on, you set up your system on this private side of the platform for documenting your methods. You don't need to decide now what's going to be published later. You can start private, you can stay private, and then, when it comes time to maybe link something to a research paper and share your methods, it's very easy to do that and move that protocol that you have on the private side of the platform into our open methods repository. And so on the public side of the platform, we are similar to a preprint server, but very specific to methods. And what you can do is when you do publish your protocol on the public side of the platform, it will receive a DOI, so a permanent identifier that other people can cite. You can link those methods to your research papers, and I'll show an example of that in a moment. And that means as well that you can get credit for the work that has gone into method development. And so we have around 25,000 public protocols that people have published so far. And you can also, peruse others' protocols and then build upon them as well. So we have functionality where you can create, for example, your own editable copy of these published protocols to easily reuse and tweak for your own research or lab setup. And so there's a lot you can do on the private side of the platform. And so you'll see an example of a protocols.io protocol there on the right. And what I want to highlight is that unlike something that can be relatively static, like a Word or PDF document, the protocols in protocols.io are very dynamic and interactive. So you can add in things like tables, plate maps, images, videos, what we call components, where you can put in amounts that are used and can be scaled up and down. We also have AI-assisted protocol drafting and troubleshooting. And this AI is trained on the Springer Nature subscription catalog of high quality methods and protocols. So the methods from that journal I mentioned earlier, Nature Methods is one of them. So this is information that is not available to tools like ChatGPT, for example. What you can also do is when you have a protocol that you have written on the platform, you can document exactly how that protocol was followed on a specific day via a run record. And you can do that via mobile, tablet, or computer. I'll show that a little bit more detail on the next slide. And then the protocols themselves, both on the private and public side of the platform, can be versioned and forked. So you can think about a version is you're updating the same method for the same purpose, whereas a fork would be you're adapting the method for a different purpose, something like a different cell line, for example. And it's maintaining a trail of how that protocol evolved. So those versions or forks are linked together so you can see what happens over time. And then the protocols are very interactive in that on both sides of the platform, you can comment on the individual steps on the whole protocol to ask questions and make suggestions. And I think I've already touched a little bit on the DOI and collaboration. This is what the run record looks like, which I was talking about just before. And so we can see here, you can see it's a little bit of a different view to what was shown on the previous slide, but you have each of the steps. And then as you're in the lab, you can mark that as done, and it's recording exactly when you've completed that step, who by, and you can add in any notes or additional information on what you've actually changed in that method on the specific day. And then you're creating a record of how that protocol was followed, which won't change the underlying protocol, but you can then use that record and export it, put it alongside any data that you've generated, things like that. So that was a little bit about the private side of the platform. But if we think about once you've used these tools and it becomes time to publish your methods, what might that look like with protocols.io? And so what we kind of want to facilitate is this concept of modular publishing. And this is where discrete research outputs are put into their own fit-for-purpose repositories and then linked to the research paper that might detail the narrative or story around that research. So you can see in this circle here, there can be lots of different modular outputs. So things like molecular structures, code, data, images, or protocols. And we have a really nice example of modular publishing here on the right. So this is a Nature Communications paper. And we can see in their data availability section, they have said all of the places we can find the relevant information. So their expression constructs have been deposited on AdGene, their protocols have been deposited on protocols.io, and their molecular structure has been deposited into the protein data bank. And so these places are all built for the different types of outputs. You know, if we think about a 3D molecular structure, you can put 2D images into supplementary information, but it makes a lot more sense to put it somewhere like the protein data bank where people can visualize it in 3D, they can download the files, and it's the same idea with protocols.io. And there are several benefits of taking this approach. One of them is that when you do have separate research outputs, you can have separate author lists. So on this slide, it's the same paper as I showed on the previous slide, and we're looking at the author lists. So we can see that in the paper, one of the authors, Kelsey Ramirez, she's fourth author, but she was a really key developer of the protocol linked within that paper. And so she can be first author on the protocol. She can list that as an output on her CV and really track um, the impact of the output that she was mainly responsible for, and also demonstrate what her contribution was there. One of the other benefits, which I touched on a little bit earlier, is that it can increase the discoverability and reuse of your research, specifically across multiple disciplines. So in this example here, we can see that there's a paper, and the subject of that paper is on three-spine sickleback parasite. So that's a fish parasite. And we saw this exchange happen on Twitter where someone was looking for an RNA extraction protocol for primary cortical neuron cultures. And then this other person happened to say, you should try this basic extraction protocol from protocols.io. And so this is the protocol that was linked to this paper. And because it was put onto protocols.io as an RNA extraction protocol, it could be findable and used by researchers across multiple disciplines. But if this was just in the paper or in the supplementary information, I think there's kind of a question of, would this researcher working on, brain cells really be looking at this paper on a fish parasite? So probably less probable that he might find that. One of the other benefits of modular publishing is that you can have separate research outputs that can have distinct access models. So even if it's not possible for a paper that you're publishing to be open access, you might not have the funding, you might not be the person making that decision. If you put your methods onto protocols.io, you can ensure that the methods are open even if the paper can't be. And this is an example here. So we have a Nature Cell Biology paper that's from a subscription-based journal. And the methods that they have linked to that paper are on protocols.io, so everybody can access them. If we zoom out a little bit, this is what a publishing workflow might look like using protocols.io. And we can see here is that you can update the protocols over time via new versions. So we know that, the science method development, it doesn't stop after a publication happens. So with protocols.io, you can update the methods that you've published after you've published a paper. And the evolution of those methods is very transparent and trackable to you and to other people who come across them. So we can see in this timeline here, this is from this protocol that's published on protocols.io. So these researchers, they published a preprint and they linked a protocols.io protocol to that preprint. And then, a little bit later in the same year, they were putting together a formal publication, 2+1. And before they did that publication, they created a version 2 of that protocol because they'd made updates and changes that were relevant to this publication. And then even after the paper came out, they made more internal improvements to that protocol, and they were able to publish those the following year. And then because that method is on protocols.io, it can still now be built upon by other people via new forks, for example. On the topic of kind of publishing new versions as well, what people do ask us is, what happens when I publish a new version? Is it the same DOI? Is it a different DOI? Is there something that I need to manage there? And so what we can say to that is that every time you publish a new version, there is a new DOI that's created. But what we have is this concept called dynamic permanence, where If someone follows, for example, a DOI to version 1 of your protocol, they'll be taken to version 1, but they will be notified that there's a newer version of that protocol available. So they can always see the version that was published, back at that time, but then they can see the improvements. And they can even go in and kind of compare to see what the differences are as well. So I hope that gave everyone a bit of an idea on how protocols.io can help you throughout all of the stages of your research. And what I will do is, if we have time, I'll pop out of the slides and into the platform and show you a protocol and protocols.io in a little bit more detail. But before I do that, I did want to highlight a couple of, you know, voices of people who've used Protocol Studio and have great things to say about it. So this is a nice quote from Dr. Jacopo Sarasoni. So he was an early career researcher and he found it really great to give him visibility and opportunities. So he published a protocol on protocols.io and then got that peer reviewed and published along with Plus One. That had a great impact for him. Similarly, another early career researcher, Dr. John Barrows, he said that publishing on protocols.io really allowed him to establish himself as someone with expertise in a specific technique. And because of that, he had people reach out to him as a reviewer and it facilitated other connections. If we look at kind of, more established PIs, so this is Professor Luciano Martellotto, and he publishes quite a lot on protocols.io, and he said that 80% of his recent collaborations have been as a direct result of sharing publicly on protocols.io. So, we get a lot of traffic to protocols.io. People are finding these protocols independently of research articles, so it can be a really great way to facilitate research connections. And so I think it's a really good time to start thinking about this because it's becoming a lot more common and thought about across policy. So if we think about the open science movement historically, it has focused on open access publications, open and fair data and code, but it's becoming talked about a lot more now that, if we're sharing data, we need the methods that go along with that data because if you want to really understand what the data is and accurately interpret it, you need to know how it's produced. And so this is a list here of a couple of policy recommendations around methods sharing. So it's becoming a lot more kind of integrated into the landscape and is really recommended by quite a few places. So you can see in this list here, so obviously protocols.io is a Springer Nature product, so we are recommended in Springer Nature journals, but even outside of Springer Nature, widely other journals recommend kind of putting your methods on protocols.io during manuscript submission. Similarly, with grant funders, um, for example, you'll see here ASAP, they are a funder. They actually say that if we're funding your work, you must put your methods onto somewhere like protocols.io. And so SciLifeLab can provide premium protocols.io for you. There's also many other institutions throughout Europe and the world who provide and use premium protocols.io. So on that note, as Joanna and Angela said, they can provide premium protocols.io to you as part of being SciLifeLab. So there are, a few benefits to using the premium side of the platform. One of them is the ability to create unlimited private protocols and files. So With the free account, you're limited to 2 private files. So it does quite limit your ability to kind of create that space to manage your methods before pushing them public. With the premium account, you can also have unlimited workspaces. So I didn't talk about that very much today, but those are the places where you would collaborate in teams and um, store your protocols. And you can kind of create unlimited spaces for different teams, different projects with the premium account. And then what you can also do is invite, so you can have unlimited SciLifeLab members within your workspaces, but if you do collaborate with people outside of SciLife, you can invite up to 10 of those into your workspaces as well. And then you'll see a couple of other things on this slide here. What I'll talk about in a moment is the protocol entry service, which you'll also have access to. So if you are interested in getting the premium license via SciLifeLab, I've put some instructions here, which I think Joanna and Angela have passed along to me. So these are the steps that you'll need to take. So start off by going to protocols.io and creating a free account using your institutional email. And then you'll need to contact Joanna and Angela and put some information in there. I think they'll talk about this in more detail after my presentation. And this is just showing you it's very easy to create an account when you do need to do so. So you go to protocols.io, click this sign up button, and then you can create an account. And so make sure you're using your SciLifeLab email address. Once you do get your premium account, these are some steps that I would recommend taking to help you get started. So I've got kind of one if you want to manage your own methods, another if you're managing them across a team. And so I think the key theme here is that start off with a couple of methods that you're already using and bring them onto the platform and start using them there. But it's really the best way to learn and use protocols.io is just to kind of get started creating something on the platform and have a play around. If you are following these steps and you do get stuck anywhere along the way, you can always ask us for help. So this link here is where you can find and submit a request for a demo. And so that will be about 30 minutes where you can chat. It will probably be me or Emma and ask any questions that you would like, and we can show you how to do things. And then this is highlighting the protocol import service that you have access to. So I mentioned before, the best way to get started on the platform is to bring onto protocols.io a protocol that you're already using day to day. And so we can help you do that. What you can do is go to this address and simply drag and drop a file. It can be, a paper, it could be a picture of your lab notebook, it could be a Word document, and then we'll have a team of editors behind the scenes who will put that into the protocols.io format for you. And just to finish off, as you're using protocols.io, if you do need any additional help, this was a very kind of introductory webinar, but we do have various places aside from the demos that you can get help. All right, so that is all from me. We do have here on the slide as well a webinar feedback survey. So it really helps us make sure that the information that we present is as useful as possible for you. So I'll put that link into the chat as well. So we'd really appreciate it if you filled that out. Thank you.
I think this is a very nice bridge to the last part of this webinar. Angela, may I ask you to share the screen again with the presentation? because we would like to present of how we envision working with you. So we have a continuation within SciLifeLab, even if some of the researchers lose connection to the group and SciLifeLab. This slide pretty much summarizes what Julia already said, that what is our purpose of having this premium license for SciLifeLab affiliated researchers. We are all about fair and open science, as you probably know, but because of the limitation of the free license, we have this service for you, so you can collaborate within your workspace and you can develop further protocols. And as Julia said, the number of private protocols is unlimited in the premium workspace. So you have this area where you can work on new methods. Of course, we promote research reproducibility, but what is quite important also for us is that you get attributed and recognized for your hard work in developing methods. So we strongly acknowledge you to share the methods. I mean, protocols is just one of the tools. It's an excellent tool, and we really would like you to join. It has a good connection to ORCID ID, so the method you publish is going to appear on your list of the publications under ORCID. But it's quite important that you get seen for the work you're doing. Next, please. Down to the point, how to get the premium license. We require SciLifeLab affiliation of any kind, so you have to message us about it. We also recently reviewed the current status, and we had 70% of users that were inactive, that just had a sneak peek in the system, never created any protocol. So we will do yearly reviews of the users, and if you don't use it at all, you will be downgraded, let's say, to the free license again. It doesn't mean that you're losing it. You can get it again, or we usually contact people before we do it. But we really would like to have in this system people who are actively working within the group. So we prefer to have this continuity. So we would like to have premium licenses and create a workspaces for PIs or lab managers or group leaders, anyone basically who with by agreement within the group hold the license. That means, as Julia also said, that if this person invites people from SciLifeLab or external collaborators to the group, they will all have the possibility to use premium features for this contact. We would try to avoid adding students to the premium license because they usually work for 1 or 2 years, sometimes even less on the protocols. So we would like the holder of the workspace to be someone who is more permanently affiliated with us. But of course, everything can be discussed. If you have a postdoc for 2 years only in your group and he will be the only person working on protocols, we can provide a premium license to this person. And well, it's for the groups to be used. We just want to have an efficient usage of the system and be able to serve as many groups as possible. Next one, please. So this already was said, but just quickly to the points. Please, before contacting us, create a free account with your SciLifeLab email address, preferably SciLifeLab.se. I know that we have a lot of others with SciLifeLab. Then contact us, tell us, this is my address, this is my affiliation, I'm planning to use it for developing new methods in the lab, or I'm planning to publish the methods that we have. One sentence, few words, really it's enough for us. And then we guide you through the next step, and you will know that you have been added to the premium license. Most likely, you will have to join the entry workspace, but we are still working on the process, and yeah, we stay in touch. Next one, please. So we prepared entry website to the service where There is more information, a lot of information that has been already provided today. And also user guidelines and workspace admin guidelines. If you create a workspace, you are an admin, that are tailored to the kind of work that we're doing in the SciLifeLab. This is the QR code going to the space. This is the actual address underneath. Please have a look, check, read. I think most of the content that is there was already communicated today. And just write us and we will answer to quotes. And we are really, really hoping that you will join the service and that you will be using it. Then you will be publishing your protocols, your methods. It's not only laboratory protocols, by the way. There is some standards for bioinformatics methods or for standard operation procedures, and that you will be recognized in the world for publishing your methods. Yes, also Emma, thank you so much. Emma also posted on the chat the link to the webinars. There are excellent webinars by protocols.io, although they are a bit more general than this one. This one was tailored for the people affiliated to SciLifeLab or working in the life sciences. Thank you very much for your participation. Please contact us, and yeah, we're looking forward to working with you.